Hello everyone, I'm Dr. Ankita Singh, Leader, Department of Oral Pathology, Bapu Banarsi Das College of Dental Sciences, Lucknow. And today I will do lecture on histochemistry of oral tissues. So what is histochemistry? It is a technique of biochemistry and histology which allows us to see the chemical constitution of cells and tissues. Like I've said, it is a branch of science which is concerned with the identification and distribution of the chemical constituents of tissues by means of stains, indicators, and microscopy. So there are histochemical stains which are available, which provide high contrast and that are helpful in image analysis. Techniques are based on precise chemical rationales for their ability to stain different biochemical substances. They provide in situ information that cannot be obtained through biochemical methods. So basically, there is localization of specific proteins and glycoproteins, etc. Application of immunobiological principles in histochemical localization of specific proteins and glycoproteins allows detection of host of biologic molecules that are needed for normal development. Then there is latest technique called in situ hybridization. It allows to identify the gene or the gene product. So in situ hybridization is the latest histochemical technique that permits identification of a gene, that is the DNA sequence, or a gene product, mRNA, on tissue section. Then histochemical techniques can be used for qualitative analysis of chemical substances in cells and tissues. This line is very important. For quantitative analysis, methods like double beam recording microdensitometry, microphotocell counter, and scanning and integrating microdensitometry can be used. Then light microscopic histochemical techniques have been developed for use in electron microscopic histochemistry. Then there are histochemical techniques, and we should know that when tissue comes to us, we should preserve it in lifelike form. So we use fixing solutions like formaldehyde to preserve the tissues. So fixation is done to preserve tissues in lifelike form. Factors which are involved in fixation are fixative solutions used, optimum osmotic conditions, cold temperature and controlled pH of fixing solutions. Like I said, formaldehyde is the most commonly used fixative. Formaldehyde is generally used as 10% solution buffered at pH 7 at cold temperature around 0 to 4 degrees Celsius. Then apart from formaldehyde, we can also use acrolein or butyraldehyde that are frequently used as aldehydes for fixation. Then Roseman's fluid is used for visualization of glycogen, glycoprotein, or proteoglycan. Carnoy's mixture are used for histochemical staining of nucleic acid. Then highly labile enzymes like cytochrome oxidase that cannot be preserved by chemical fixation are fixed by freeze drying. Study of teeth and bone for light and electron microscopic histochemistry requires simultaneous fixation and decalcification with formaldehyde and EDTA. Then there are specific histochemical methods that can be categorized, and they are glycogen, glycoprotein, proteoglycan methods, then protein and lipid methods, then enzyme methods. Methods for glycogen, glycoprotein, and proteoglycan. FAST technique is most commonly used for detection of carbohydrate groupings. Proteoglycans can be demonstrated by thiazine dyes like toluidine blue. Toluidine blue is a metachromatic dye, and azure A and Alcian blue. Proteins. Histochemistry of proteins is based on classic reaction of amino acid groups. Then lipids, for lipids we use Sudan dyes. So total lipids are studied using fat colorant dyes like Sudan dyes. Chromation of formol, calcium fixed tissues, and staining with Sudan black is used for identification of phospholipids. Enzymes, we use Gomori method for phosphatases. It uses phosphoric esters of glycerol, glucose, or adenosine. Coupling azodase technique, they are used for demonstration of phosphatases. Then azodase techniques are better suited for phosphatases in teeth and bone. And then immunohistochemistry. Immunohistochemistry is based on antigen-antibody reaction. So precise localization of specific biologic molecules in the intracellular compartment, on cell surface, or in extracellular matrix can be done. Like I've said, in immunohistochemistry, they take advantage of antigen-antibody affinity through its ability to identify and localize proteins of interest by detection with labeled conjugates. So that we use fluorescent dyes. Enzyme histochemistry. Enzyme histochemistry demonstrates the activity of enzymes which are present on tissues. So for visualization of this, we see the action of enzymes on a specific substrate. So following this reaction, an insoluble product is formed which provides the location of the enzymes. So basically in enzyme histochemistry, we see the location of the enzyme. The technique of enzyme histochemistry, which relates structure and function, can be used to locate many enzymes, including acid phosphatases, dehydrogenases, and peroxidases. An enzyme indicate what is happening in the tissues. Now, histochemistry of oral hard tissues, carbohydrate and proteins. PASS method, most commonly used to study composition of teeth and bones. Normal, mature bone and teeth, no reactivity with PASS technique. Then developing, resolving bone and dentine, they intensely stain with PASS. Interglobular dentine, poorly calcified dentine, and odontomas, there is clear PASS reactivity. Enamel matrix, non-reactive with PASS. Now, specific methods that identify amino acid groups in proteins are DNFB and ninhydrin-skip method. DNFB is dinitrofluorobenzene. So, absence of carboxyl and amino complexes in predentine and osteoid and their presence in dentine and bone can be revealed by histochemical techniques. Then there are phosphorin, which are present in dentine and odontoblast, and they can be identified by histochemical techniques. Then enamelins are there, aminogenins are there. They can be identified by histochemical techniques and using monoclonal antibodies. Then lipids, they are demonstrated by sudanophilic reaction in the odontoblastic process and enamel rod sheets. Sudanophilia, it is widespread in developing tooth, in the zone of mineralization also, in predentine also, and in basal zone of ameloblast. Enzyme histochemistry of hard tissues. We have alkaline phosphatase, which is seen in areas of mineralization. It is associated with osteogenesis, dentinogenesis. Osteoblast and odontoblast stain positive, and it is seen in stratum intermedium, Koff's fibers, and ground substance. Adenosine triphosphatase, which is also called ATPase. Calcium-dependent ATPase, they are seen on distal and lateral cell membrane of ruffled and ameloblast. In smooth-ended ameloblast, it is seen along basolateral regions. So the enzyme controls the access of calcium into the developing enamel matrix. Then there are acid phosphatase and esterases. Acid phosphatase, they are localized in lysosomes, osteoclast in bone, and odontoclast in dentine. And esterase, use of specific naphthol esters have revealed an intense staining reaction in calcifying matrices of bone and dentine. They're also seen in cells and microorganisms associated with calculus deposit in teeth. Then there are cytochrome oxidase. Its histochemical localization reflects oxygen requirement of cells and tissues and their level of metabolic activity. Osteoclast, osteoblast, and stratum intermedium of molars and incisors show oxidase activity. Histochemistry of oral soft tissues. There are polysaccharides present. So for that, PASS method is used. Then epithelial glycogen, they increases during repair and inflammation. The non-keratinized mucosa are there. They reduce constant level of glycogen. Benign and malignant epithelial proliferation, they show increase in glycogen. Proteoglycans with chondroitin sulfate and hyaluronic acid, they are the major component of human gingival epithelium. Then there are heparin and histamine. Heparins, they are present in mast cells and they are stained by metachromatic stain like toluidine blue. And histamines are there, they can be demonstrated by fluorescence microscopy. Then there are proteins and mucins. Proteins like keratin is stained Prussian blue by ferric ferrocyanide method, and mucins, salivary mucins, they can be stained positive by mucicarmine. PASS stains neutral mucins. So that is all for your lecture. Thank you.